embryonic stem cell research is still in its infancy, and its potential should be understood in this context. Human embryonic stem cell studies are in early stages and are currently being used to help understand the differentiation and functions of many human tissues, including neural, cardiac, vascular, pancreatic, hepatic, and bone. Embryonic stem cells have two properties that make them promising for research in cell therapy. First, they can be grown in tissue culture and therefore provide an abundant, renewable source of cells. Second, they are pluripotent and can, theoretically, be directed to become virtually any type of the adult body, any cell type in the adult body. But while much has been postulated about the ability of embryonic stem cells to generate transplantable cells for medical applications, many scientific hurdles remain before they could be used clinically in the United States. One of the difficulties specific to embryonic stem cells is their propensity to form tumors. This dangerous property is the corollary to their extensive growth potential. Other issues needing solutions include questions of how to generate functional differentiated cells and how to solve immune rejection issues. Although there is a high potential for treatment of degenerative diseases with human embryonic stem cells, development of practical application lags behind adult stem cells and indeed has been limited to animal studies thus far. Advances in alternative sources of pluripotent cells. Recent biological advances have raised encouraging possibilities for producing powerful stem cells without harming human embryos. What scientists value most about embryonic stem cells is their pluripotency and expandability, that they have the potential to be teased into many and perhaps all of the different cell types in the body. But scientists have begun to find out that this potential may also exist in certain cells derived without embryos. New ways of producing pluripotent cells that don't require the destruction or even the endangerment of human embryos are now being investigated. In May 2005, the President's Council on Bioethics published Alternative Sources of Human Pluripotent Stem Cells, a white paper which suggests 4 possible approaches for alternative sources. 1. By extracting viable cells from embryos already dead. 2. By non-harmful biopsy of living embryos. 3. By extracting cells from artificially created non-embryonic cellular system or entities engineered to lack the essential elements of an embryo. And 4. By reprogramming or dedifferentiation of adult stem cells back to pluripotency. Each of these methods carries its own scientific and ethical uncertainties, but one or more may ultimately offer a plan towards an ethically responsible source of pluripotent stem cells. Very recently, landmark research from Wake Forest University has suggested yet another alternative, amniotic fluid. Investigators discovered contains highly flexible stem cells shed by the fetus. These appear to have all the valuable qualities of embryonic stem cells, plus some advantages of their own, like greater ease and speed of culture, and no tendency to produce tumors. Meanwhile, they are comparatively easy to collect without harming human life. We begin our review with the 4 methods identified by the Bioethics Council. Just in the short period since they were sketched out in May 2005, significant progress has been made on each of these techniques, as reported in a number of new peer-reviewed research studies published in leading scientific journals. At this point, one of the most promising avenues appears to be somatic cell reprogramming, which uses chemical and genetic factors to reprogram an adult cell to function like an embryonic stem cell. Each cell in an individual's body has the same DNA as every other cell, but in the course of developing into specialized adult tissues, different cells undergo different patterns of gene activation. Somatic cell reprogramming seeks to switch on or off the appropriate genes to transform an adult stem cell back to the equivalent of an embryonic stem cell. This might be accomplished by stimulating the adult stem cell with the right combination of chemicals and genes, or by exposing it to the cytoplasm of an existing line of stem cells. This method could, in theory, create stem cells in bulk while bypassing entirely the problem of creating and destroying embryos. 2 research teams that have demonstrated significant progress towards this sort of cell reprogramming are Kevin Egan and Chad Cohen from the Harvard Stem Cell Institute, and I apologize if I mispronounce these names, Shinya Yamanaka and Kazutoshi Takahashi from the Department of Stem Cell Biology in Japan's Institute for Frontier Medical Science. In August of 2005 and August of 2006, respectively, each team published impressive results, which seem to produce pluripotent stem cells by reprogramming ordinary adult cells. The project conducted at the Harvard Stem Cell Institute fused a human adult stem cell with an embryonic cell, and these could come from one of the embryonic stem cell lines that President Bush has approved for use with the federal funds. That effectively turned back the clock on the adult cell such that it was reprogrammed to a pluripotent state. Egan and Cohen believed their research could lead to an alternative route for creating genetically tailored human embryonic stem cells for use in the study and treatment of disease. Doctors Yamanaka and Takahashi published research based on mouse cells. Their complex study produced stunningly simple results. They reprogrammed adult stem cells into a pluripotent state simply by bathing them in 4 genetic factors. The finding is an important step in controlling pluripotency, which may eventually allow the creation of pluripotent cells directly from somatic cells of patients, comments Dr. Yamanaka. If successful, this could offer all the benefits of embryonic stem cells and more. These cells could be genetically matched to any prospective patient without the ethical dilemma of embryonic destruction. Other promising avenues are likely to be open in the future. The latest alternative published on January 7, 2007 in Nature Biotechnology involves amniotic fluid stem cells. Dr. Anthony Atala and a team from the Institute of Regenerative Medicine at Wake Forest University School of Medicine and the Harvard Medical School reported on a new category of already available stem cells extractable from the waters cushioning the babies in utero, as well as the placenta. While this very new research will need to be replicated and confirmed, Dr. Atala and colleagues have already managed to grow useful brain, bone, liver, muscle, and other replacement tissues using these stem cells. This discovery suggests that if all U.S. newborns had their amniotic stem cells frozen, they could be made available for future tissue replacement without fear of immune rejection. Moreover, a bank of amniotic stem cells from the waters of 100,000 pregnancies could supply 99% of the U.S. population with genetically compatible stem cells for possible transplantation. More studies will be needed to confirm that amniotic stem cells can generate all other cell types, but so far, every culture attempted has succeeded. It must also be determined that tests conducted in mice will translate into humans. Meantime, that is but one of several promising glimmers in the latest research. What follows is a summary of the preliminary analysis of the Bioethics Council on the 4 alternative approaches they discussed in the May 2005 white paper, supplemented by fresh additional findings from the most significant peer-reviewed studies published in each area since May 2005. It is important to note that while many of these alternative approaches show great promise, all of them are still being tested in animals and are thus in the early stages of development. The Bioethics Council has conducted only a preliminary analysis of the approaches as used in animals. The administration does not endorse any of these specific avenues of research in humans at this time. All new approaches will have to be carefully considered by scientists, regulators, and others as appropriate on a case-by-case basis as further information and research becomes available. It may be that some of these research proposals could prove inconsistent with the Dickey-Wicker Amendment protections against creating, destroying, or harming human embryos, or they may fail some other test of ethical or science viability. But the cumulative weight of this emerging science is, as a Washington Post Science journal recently summarized, adding credence to an emerging consensus among experts that the popular distinction between embryonic and adult stem cell is artificial.